now time to study cells. Cells, of course, you know, are the basic unit of life. Okay, the cell theory states cells can only come from cells. Cells are the basic unit. Cells take in, acquire energy, and use it, and they carry the genetic material. Okay? Before we get into the two basic types of cells, I want to just kind of show this little diagram, which is interesting, I think. We'll get into the origins of cells a little bit more later on, but we now have, you've probably heard of 5 kingdoms. Actually, there's 6 kingdoms now, and actually 3 domains. So 3 domains is the higher order of classification, encompassing everything. Okay, so for example, all organisms share certain things. We all have a genetic code made up of nucleotides. So we either have DNA or RNA, and the genetic code is the same. So we'll get into the genetic code later on, but it's the same for all organisms. Okay, and of course, everything is made up of cells. Right? So if you look back about 3.5 billion years ago, we all had this common ancestor. Okay, and from that, and we go up here, we get a split. Okay, so we get a split between the monairans or bacteria. Okay, one-celled prokaryotes. Okay, and then it branched off to become eukaryote, but off of that branch becomes archaea. Okay, archaea, we'll study those a little more later on, but those are pretty thought to be more similar to bacteria earlier on, but even though they are prokaryotic, they actually are more similar to eukaryotes. So this is the kind of a real basic tree of life here, which we'll look at more later on. Okay, but back to cells. Okay, cells, there's 2 basic types of cells. All right, first one are prokaryotic. Okay. So prokaryotic include the monairan and the archaea. I don't think I spelled that right, but it's close enough. They are single-celled organisms. Okay, small. This is not drawn to scale. Okay, they're really small, single-celled organisms, and they have certain characteristics that are different than eukaryotes. But one thing they do share is that they both have ribosomes. But let's go back to DNA first. Okay, their DNA is organized in 2 different ways. Okay, one is just a big, long, circular DNA strand. So that's here. So for circulars, you know, by that I mean it's connected. Okay, so that's how their DNA is organized. Again, DNA, it's got the bases A, T, G, and C. Okay, same code as us. But they also have these smaller DNA strands are called plasmids. Okay, so this is how bacteria, quote, get variety or have sex. They actually exchange these plasmids between different bacteria. And it doesn't have to be the same species. It can be different species of bacteria, but they can exchange that. Of course, the problem there is if they have, a lot of times you carry resistance to an antibiotic on this little plasmid. So if this is, say, gonorrhea, which has a resistance to an antibiotic like penicillin, they can transfer that to other bacteria, staph infections and so forth. So that's a problem that medicine has to fight. All right, but that's how DNA is organized. They all have ribosomes. Of course, ribosomes is the site for protein synthesis, okay? But they are free within the cytoplasm. Okay? That's where the proteins are produced. Okay? They have, of course, a cell membrane or plasma membrane, and outside that they have a cell wall. Okay? And then outside of that they have these pili. Okay? Not all of them do, but a lot of them do have this pili, which little hair-like structures. Used for movement and for adhesion. They have a lot of glycolipids, which allow them to stick to different things, and then a lot of them have a flagella for moving. So those are basic characteristics of prokaryotes. Eukaryotes are more complex, so to speak. They have membrane-bound organelles. There's 4 different kingdoms that are eukaryotic. Okay. Protista are single-celled. The rest are multicellular. So funguses, plants, and animals are all multicellular organisms. Okay, so we have a specialization of function within the cells. Okay, so if I draw a base cell like this actually happens to be a plant cell. That cell can vary according to whatever function it has. Okay, so. For example, in animals, some cells are really long and skinny, like a nerve cell or a neuron, or a muscle cell. Those are long and thin, whereas a fat cell would be more spherical. Okay? So depending on the function, skin cell would be, of course, flat, epithelial cell. Okay, so they can be specialized. Okay. But here are structures, okay, or organelles. A lot of them are membrane bound within a eukaryotic cell, which all, of course, have a certain function. Right? So, of course, a nucleus. Nucleus is membrane bound. So in this little diagram, it's here. Again, not drawn to scale. Of course, inside the nucleus, that's what harbors the genetic material. Okay, so we have a nucleolus inside here, which helps make the ribosomes. But also we have these chromosomes, which are linear. The DNA is organized linearly. So unlike prokaryotes, which is circular, these are linear. For example, in humans, we have 23 pairs of those. So there would be 46 chromosomes in a somatic or body cell. And then, of course, we have these ribosomes, which are site of protein synthesis. Okay? The ribosomes can be free in the cytoplasm, okay? Or they can be on the endoplasmic reticulum. So let's move to the endoplasmic reticulum. Okay? It can either be rough or smooth. Okay? So rough endoplasmic reticulum has, of course, ribosomes on it. So that's where proteins are made. So proteins are made, and then the rough ER is used to then transport those proteins, okay, and modify them a little bit. Okay, smooth ER, okay, right, like this. They're actually for lipid synthesis. Okay, so they don't have the ribosomes, but they're responsible for making lipids. Then Golgi Apparatus. Golgi Apparatus are again in the cytoplasm there. They are used. They generally pick up vesicles, okay, that are pinched off from the endoplasmic reticulum or the smooth endoplasmic reticulum. Okay, they some little vesicles here, so they engulf them, modify the proteins or the lipids, and then transport them where they need to go. Okay, so Golgi apparatus is used for picking up, modifying, and transporting proteins. All cells have mitochondria, whether it's a plant cell or an animal cell. They have mitochondria. Of course, the mighty mitochondria here. It's a double membrane system. Okay, so you have an outer membrane, and you have an inner membrane, which is called the cristae. Okay, and inside that, you have the matrix. Okay, this is, of course, where energy is produced. Then you have chloroplast, if it's a plant cell. Okay, chloroplast actually is a triple membrane system. So you got an outer membrane, inner membrane, and then you have inside membranes here, where the it's called the thylakoid membrane. That's where light is actually captured. Okay, so light energy comes in and excites the chlorophyll molecule, and we take that CO2 and oxygen, or excuse me, CO2 and water, and make glucose and oxygen from it. And we'll get to that, of course, more later on. Okay, so there's the chloroplast, and vacuoles. Animal cells and plant cells have vacuoles. In plants, it's really prominent. Okay? So you have a large central vacuole. Of course, the plants are mostly for storing water. You can store other things. But of course, if a plant is dehydrated, this shrivels up. Okay? And that shrivels up, you get what we call maybe a plasmolysis, and a plant will just show outward signs of wilting. If you give it water and it's not too far gone, then this will store up water and it presses up against the membrane and then against the cell wall, and that causes what is called Turgot pressure, and makes that plant, gives that plant structure, allows it to thrive. Okay. Of course, outside that you have a cell membrane, you have a cell wall here. Also in plants have lysosomes. Okay, they're just small circular. It's hard to distinguish just by looking at it. The difference between a lysosome and a vacuole. Essentially, lysosomes have hydrolytic enzymes within them. Those hydrolytic enzymes, of course, do hydrolysis, so they chop up things to smaller molecules. Some is just breaking up toxins, and some is for actually breaking up molecules to recycle them, so get them back down to the monomers, so then they can be used by the cell to make new macromolecules. All right, so there's your short